So moving on to our second talk, another excellent offering, this time Sarah, who's going to be talking to us about global characterization of modeled micronekton. So over to you, Sarah, with thanks.
Okay. Good afternoon. Can you see my screen?
Yes, it's on slide sorter at present. Now it's on full screen.
Okay, great. So my name is Sarah Albernhe. I'm a PhD student in France between CLS and DELOPS. I'm very happy to be here tonight, so thank you for this opportunity to talk about my work. I'm studying micronekton modeling, and today I'm going to talk about global characterization of modeled micronekton in biophysically defined provinces. So this talk is based on a paper that has been published a few weeks ago. So you can scan the QR code to access the online article if you're interested. During this talk, I'm going to introduce micronekton quickly for those who are not familiar with it. Then I'm going to explain how I defined the biophysical biomes, how I characterized them with modeled micronekton biomass, and how I validated the boundaries between the biomes with acoustic data. And I will end up with a comparison with other regionalizations. Sorry. It's not working the way it should. Okay. So micronekton is a group of marine organisms from 2 to 20 centimeters. So we have cephalopods, small fish, crustaceans, etc. And they are located at the intermediate level of the oceanic trophic web. So they feed mostly on mesozooplankton, and they are the main prey of marine predators like tuna. And the very interesting behavior about micronekton is its diel vertical migrations. So it migrates every day between the surface and the deep ocean. Basically, it remains in the deep ocean during the day to hide from predators, and it goes back at the surface during the night to feed. This migratory signature can be observed on acoustic transects. So in this study, we are going to consider acoustic backscattering as a proxy of micronekton biomass. And performing those diel vertical migrations, we know that micronekton is a key player in the biological pump as it exports and sequestrates carbon to the deep ocean. So that's one of the main reasons why we are interested in micronekton modeling. Together with its central position in the marine trophic web, which represents a huge interest for fishery science. And so in this context, why did we want to define biophysical biomes? The idea is to investigate if micronekton homogeneous functioning patterns can be identified using a very simple set of biophysical variables. So we considered 3 environmental variables that are known to have an impact on micronekton. So we had the epipelagic layer temperature, the stratification of the mesopelagic ocean, and the net primary production. To define these biophysical biomes, from these 3 environmental variables, we performed a clustering using the machine learning algorithm K-means. And we applied the learning phase of K-means on data of these 3 environmental variables averaged from 1998 to 2019. This way, we get a definition of static reference biomes, which are very representative of the average state of the ocean during this whole period. Once we have these reference biomes, we project the clustering on monthly data over the same time period. We apply this time the prediction phase of K-means algorithm, and so we get a monthly time series of biomes from 1998 to 2019. So that was the very quick methodology. Now let's have a look at the results. Here you can see the map of the reference biomes, so obtained by the clustering of the 3 environmental variables averaged over the whole time period. We defined 6 biomes, as you can see with the 6 colors on the map. So we have a tropical, subtropical, eastern boundary coastal pooling systems, oceanic mesotrophic systems, subpolar, and polar biome. We divided each biome into provinces according to the ocean basin and hemisphere. So for instance, the biome number 5 that you can see in green, which is the subpolar biome, is divided into 3 provinces, 501 in the North Atlantic, 502 in the North Pacific, and 503 in the Southern Ocean. And applying this segmentation on the 6 biomes, we get a definition of 27 provinces. Okay, from this static reference definition, we project the clustering on monthly data. Okay, it works. To get this monthly time series, this dynamical definition of biomes and provinces in time. So that's based on this dynamical definition that we are performing all of our characterization, diagnostics, and validation that I'm going to talk about just after. So first, we wanted to characterize these provinces with modeled micronekton biomass. So basically, the idea is to investigate if the provinces that are derived only from environmental variables can display consistent patterns in terms of micronekton biomass. To do so, we are using the outputs of CPODIM LMTL, which I'm going to describe here quickly. So CPODIM LMTL for low and mid trophic levels. This model considers a simplified trophic chain in 4 levels. So phytoplankton, zooplankton, micronekton, and predators. And the LMTL part of CPODIM focuses on zooplankton and micronekton. It's forced by the NPP, the euphotic depths, the temperature, and the current. And in this model, we consider a discretized water column in 3 pelagic layers computed based on the euphotic depths, and that are meant to represent what you can observe on acoustic transects. So we will have an epipelagic layer at the surface, layer 1, and then layer 2 and 3, upper and lower mesopelagic layers. And between these 3 layers, we consider 6 functional groups of micronekton according to their migratory behavior. So that's the figure on the right-hand side. For instance, we have the functional group 1.1 that you can see in green, which is a resident group of the epipelagic layer. So it stays in the layer 1 during day and during night. Then we have migrant groups, for instance, the group 2.1 that you can see in red, which will migrate between layer 2 during the day and layer 1 during the night. And this way, we have 3 resident groups and 3 migrant ones. And from these 6 kinds of micronekton biomass, we will derive a characterization diagnostics for our biomes. So this one focuses on the vertical structure, really, of the micronekton. We will consider 2 ratios of functional groups from the outputs of CPODIM. We will consider migrant micronekton over epipelagic, that you can see in blue in the figure, and mesopelagic over epipelagic, that you can see in orange. In the x-axis, you can see the 5 biomes that we are going to characterize. We chose not to work with the polar biome, biome 6, in this diagnostics, at least, because the NPP that is used in CPODIM in high latitudes is different than the one used at other latitudes. So it does not allow a meaningful comparison. So we'll focus on these 5 biomes here. And what we can observe is that the box plots for the 2 ratios show different shapes and different ranges between the 5 biomes. Every pair of biophysical biome displays statistically significant differences in data distribution for both ratios, which is nice, except for biome 4 and 5 for the migrant over epipelagic ratio. It's the only pair of biome that doesn't display significant differences. So the main insight of this characterization diagnostic is that we have a vertical structure of modeled micronekton, which is specific to each biophysical biome and different from one biome to another. And so the environmental variables that we used in the clustering managed to identify areas with specific model micronekton vertical structure. We performed other diagnostics based on the outputs of CPODIM, which we can see, which you can have a look at in the paper. I would like to spend more time now on the validation of the boundaries between the different biomes. We used acoustic data to do so, and we consider acoustic backscattering as a proxy of micronekton biomass. And so the idea here is to identify potential vertical structure reorganization in acoustic backscattering between adjacent biomes crossed by a transect. We used a global database of more than 400 transects at 38 kilohertz. So you have the map on the right showing the global database. And we studied two validation metrics. So we wonder if the acoustic backscattering distribution is different between two adjacent biomes crossed by a transect, and also if it's more homogeneous within each biome. We didn't consider backscattering directly because of the many biases that are well known in comparing acoustic backscattering and micronekton biomass. We studied the proportion of acoustic backscattering received from each of the three pelagic layers of CPODIM. The map that you can see here represents the biophysical biomes for the month of November 2012, as an example. And you can see plotted above in the Indian Ocean two transects that have been recorded during the same month of November 2012. And the color of the acoustic transect represents the proportion of signal which is received from the epipelagic layer over the entire backscattering received on the water column. And what we can observe here is that on the orange biome, we have a proportion of acoustic signal received from the epipelagic layer that ranges between 60 to 80%. Whereas when we cross the boundary to the yellow biome, it ranges more between 10 to 20%. So that's the kind of reorganization that we are interested in and that we want to quantify with the two validation metrics that I mentioned. Let's go a bit deeper on these two metrics. The first metric is the between biome acoustic heterogeneity. So we want to observe the difference in the distribution of acoustic data between two biomes crossed by a transect. To do so, we apply Mann-Whitney statistical tests on transect crossing boundaries. We apply these tests distinctively on night data and day data, and also on the three pelagic layers of CPODIM. And the main result is that in 93.8% of the cases, the distributions are statistically different between the two adjacent biomes. So that was nice, but now we know there is differences between the biomes in the acoustic backscattering. We also want to know if it's more homogeneous within one biome than over the whole transect. So that's the within biome acoustic heterogeneity test. We want to compare the variance of the data over the whole transect with the variance of the data within each single biome crossed. And the main reason is that in 61.8% of the cases, the variance is weaker for the data within a single biome than over the whole transect. So the main insights of this validation is that our regionalization with environmental variables identifies different vertical distribution patterns between adjacent biomes and also homogeneous areas in terms of backscattering vertical structure. The last step of this study was the comparison with other regionalizations. So the idea is to identify similarities and discrepancies between different regionalizations that are linked to micronekton, but that are based on different approaches. So first, we defined another definition of biomes, which we will call micronectonic biomes. And basically, we applied the very same methodology than the one used for the biophysical biomes, but instead of using environmental variables in the clustering, we used the outputs of CPODIM LMTL, so we used modeled micronekton. The two variables that we used in the clustering are ratios of modeled micronekton biomass. We studied migrant over epipelagic and mesopelagic over epipelagic, average over 1998 to 2019. And we applied really the same methodology to define these 6 clusters that you can see in color in this map, in the background map. In the foreground, in black lines, you can see the biophysical biomes, so the one that I was talking previously. The idea is obviously to compare the two regionalizations, but also to compare them with the acoustic biomes defined in ARISA 2022. They are ecobiomes, so derived from a clustering of backscattering linked to environmental variables. So, what can we observe here? The biophysical biomes capture most of the patterns associated with micronekton. So we have kind of a good agreement in latitude poleward 30 degrees, so North Atlantic, North Pacific. The main temperature fronts in Southern Hemisphere, for instance, are well reproduced. The dynamic regions, so the Gulf Stream, Kuroshio, Southeastern Australia, South America, and to some extent, the productive areas and the equatorial and eastern boundary system, which means in these regions, specific characteristics of micronectonic ecosystems from model and from data are well explained by the biophysical variables. But what arises quickly is the important differences that we have in the tropical band. So the micronectonic biomes and acoustic biomes display east-west structures that identifies gyres and oxygen minimum zones, for instance, which are really not reproduced by our biophysical clustering. These geographical patterns are characterized by specific oxygen concentrations. And so this gives information on the explainability of micronekton vertical structure and biomass densities by our environmental variables. And this raises the question about the relative importance of different environmental variables in micronekton modeling in general, not only in the study, particularly the case of oxygen, because our main conclusion in the paper was that we should consider the oxygen in the clustering, and we should consider it more when talking about micronekton, maybe. And now the messages you need to take home. Our ambition is to identify micronekton homogeneous functioning patterns from a simple and parsimonious set of biophysical variables. We identified 6 biomes at global scale. We characterized them with modeled micronekton from CPODIM and MTL. And we showed biome-specific biomass and vertical structure of modeled micronekton, different from one biome to another. We also validated the biome's boundaries with acoustic data. The regionalization highlights homogeneous areas in terms of backscattering vertical structure and different vertical distribution patterns between adjacent biomes. And to end up with, the environmental variables used can account for only some of the variability of the micronekton structures, but we should also consider oxygen as a key forcing. I would like to thank the co-authors of this work. If I have a bit of time left, I would just like to show you the very first, the extension of this work that we did with the same team for the Ocean State Reports, OSR9. We studied the evolution of the biophysical provinces in time between 1998 to 2023. So we studied the trends in the evolution of the surface area and the evolution of the province's position. So on the left, you have the trend in the evolution of the province's surface areas. Basically, the provinces that are in shades of red are the ones that are going to extend, and the ones that are in shades of blue are going to shrink. So the main message here is that we have an overall decrease in the surface of productive provinces, and some of the polar and subpolar ones in favor of the expansion of equatorial and tropical provinces. Now, for the trends in the evolution of the province position, we basically studied the mean latitude of the provinces. The ones that are in shades of red are the ones that are going to drift polewards, and the ones that are in shades of blue are drifting to the equator. And so we're not very surprised, but most of the provinces are drifting toward the poles. That was the two conclusions of this work. Thank you very much for your attention. I don't know if you have time for questions, but I'll be glad to answer your questions.
Excellent. Thank you very much, Sarah. Another excellent talk. So we have actually a few minutes left. So if people have questions, and please type them into the chat or just raise their hand. While people are thinking, I've got a question, if you don't mind. So when you see the biomes expanding or contracting, is that due to organisms moving, or is it just due to the populations within those biomes shifting, so less abundant ones become more abundant and more abundant ones become less abundant?
We didn't have valuable insights on how the populations behave within our provinces. They basically go this way because mainly on the temperature and the stratification, like if we talk about the clustering, but it seems that the populations are kind of following their temperature range. I've seen several studies going in this way. So I guess as the temperature increases in like latitudinal way, I guess they'll go the same way.
Okay, but there's
I think I've lost you, Adrian. I don't know if it's the same for everyone.
no. Can you hear me okay?
Yes, I can hear you back.
Sorry, my connection broke up. I was
Okay,
just
so I
sorry.
didn't hear the question.
Sorry, I was just asking, in terms of the model, there is no horizontal migration in response to like gradients in temperature, which cause organisms to move horizontally. It's a local response. Yeah.
No.
Okay. Thank you very much. And sorry for the connection broke up. I did warn you that my connection is. So we got any other questions or comments from other people in the audience? I was going to say it's often the case that things occur to people after the talk, in which case please do feel free to email Sarah and we'll be putting the talk up on the web. But I say if anybody has any questions or comments, then please just raise your hands or type into the chat. No? Okay. It's always the way. Sometimes these things occur to people afterwards. So thank you very much to both of our speakers today, to Manu and to Sarah. I'll say I'll be putting the talks up on the YouTube page very soon, people to take a look, and please, do forward them to people who might have missed the talks. And Manu and Sarah, if there are things that you want me to include in the descriptions of those, then please do. We'll be sending around an email very soon, hopefully describing the two talks for December. Otherwise, thank you all very much for attending the talk. Thank you for our two speakers, and I will see you in the new year because the next call is being done in Australia. So see you then. Thanks very much.
Thank you.
Bye.